So I would say let's get started so that we also have time in the end, right?
Yeah,
And
absolutely.
so welcome everybody to another round of the Cancer Neuroscience seminar series. And this is my great pleasure to welcome um, Humsa Venkatesh today from Boston. And I think it's very clear we have a very pioneer of the cancer neuroscience field here with us today who will share amazing work. And just a few remarks about her CV. So Dr. Venkatesh's research studies focus on the reciprocal interactions between the nervous system and cancers. Her work emphasizes the electrical components of tumor pathophysiology and highlights the extent to which neuronal activity controls and facilitate disease progression. This is obviously a very new emerging treatment option of these tumors to really electrically disintegrate them from the neuronal circuitry. Humsa Venkatesh, Dr. Venkatesh received her undergraduate degree in chemical biology from the University of California and her PhD in cancer biology from Stanford University. She's now starting a cancer neuroscience research program as an assistant professor at the Brigham and Women's Hospital and Harvard Medical School. She has received multiple awards for her outstanding work, published multiple times in journals such as Nature and Cell. And again, it's my distinct pleasure to welcome you here today. And the stage is yours.
Great. Thank you so much, Varun. I'm really excited to be here today. So thank you both, Varun and Frank, for the invitation. I think that's such an exciting time to kick off this, cancer neuroscience concept. And I know you guys have started a series, we've started a series. And I think as we just saw earlier this week, there's starting to be a lot of different centers and hubs across the nation. So I think it'll be really exciting to see where the field goes. So today I'll be talking a little bit about my past work, focusing on cancer neuroscience in the context of primary brain cancers, and then end with some of my newer work that really extends this concept and this biology to non-glial derived cancers, both in the context of brain metastases, as well as a little bit of work that I'll show you that's very new in cancers outside of the brain. So with that, I'll just start by just giving a brief introduction about these high-grade gliomas, which this audience likely knows. But what we do know is that these devastating tumors represent the leading cause of CNS-related tumor deaths in both children as well as in adults. And though they represent 120 clinically and molecularly distinct diseases, and we've really been able to understand the molecular landscape of these tumors quite a bit over the past few decades, what's quite notable is that it's really translated to still very limited targeted therapies in the context of the clinic. And so, in thinking about when I started my training, what I really was emphasizing and how I wanted to study this disease was really a more comprehensive perspective of understanding this disease, studied in a way that not only allowed us to think about these cell intrinsic factors, these genetic and epigenetic factors that may contribute to malignant transformation, but also these cell extrinsic factors or these microenvironmental dependencies, perhaps, that inform overall tumor progression. Oops, I'm sorry about that. And of course, in the context of brain tumors, this includes a number of glial cells, vasculature cells, immune cells, together of which comprise the sum of the tumor ecosystem. And all been studied in some context in the context of the tumor microenvironment. But interestingly, what we focus on is the role of neurons and neural activity, which is kind of why we're all here today, which is a seemingly obvious component of brain cancers, but has been vastly underappreciated until recent years. And so just to give you a sense of how important these nerves are, in the tumor microenvironment, this is a clarity video of a primary patient tumor sample. And you can see that even within the core of the tumor, there is vast density of these axonal nerve fibers. And I think now this is something that we can really appreciate given a lot of our past few years and a lot of amazing work that's been done in the field. But I think this is really profound given that even just a few decades ago, it was thought that gliomas actually secreted glutamate in a neurotoxic fashion that actually killed off all the neurons in order to make space to grow. So this now understanding that they're actually a very critical component of the microenvironment is one that I think is quite fascinating. So I always like to think about this as kind of analogous to a tree or a weed of sorts. So as opposed to plucking off these individual leaves or cancer cells with these targeted therapies, perhaps another synergistic way of trying to come out attacking this disease is really pulling up those root networks, right? Preventing these tumors from grabbing these nutrients from the soil. And in a sense, we like to think about these nerves as kind of these roots that these tumors may have in their microenvironment. Now, a large number of studies really point to this idea that high-grade gliomas are thought to arise from cells along this oligodendroglial lineage, which are the cells that give rise to the myelination in the developing brain. And most recently, of course, beautiful work by Mario Suva and Mario Philbin have really shown this at the single cell level. And so these molecular and phenotypic parallels between these oligodendroglial lineage cells and these malignant versions of the glioma cells really allow us to take cues from gliogenesis and now apply them to the malignant context, or gliomagenesis. And so we tied together these microenvironmental dependencies and the developmental parallels between these OPC cells and these glioma cells to really understand these microenvironmental interactions that may be influencing high-grade glioma growth. As I mentioned, particularly thinking about nervous system. So when I joined Michel Mange's lab, they had actually just shown using novel optogenetic techniques that physiomimetic neuronal activity heavily influences oligodendroglial cell dynamics, and that with elevated activity, you could actually see an increase in these proliferation of these oligodendrocyte precursor cells, as well as differentiation, oligodendrogenesis, and this active remodeling of the myelin sheaths within that active circuit. And so, of course, as a cancer biologist, the question then became, does neuronal activity affect glioma proliferation? Now, this is a question which at the time had never directly been asked, but interestingly, a histopathological hallmark of gliomas has been found, which is this perineuronal satellocytosis, and shows these tumor cells clustering around these neuronal cell bodies. And that really suggests that perhaps in addition to a structural or functional relationship here, there may in fact be a true functional relationship, as well as a structural relationship. There is also a functional relationship here. So again, to probe what was happening here, we similarly turned to optogenetics. And just as a brief overview, optogenetics is really this technique that uses these light-sensitive opsins specifically to modulate our neural populations of interest. And here we're able to use expression of these opsins to allow for stimulation of this motor cortex that then gives us this complex physiomimetic output. And we then were able to query the response of these tumor cells specifically in response to that elevated activity. Now, what we found is that in response to activity, we did indeed see this increase in proliferation as measured both by Ki67 as well as EDU. And overall, over the course of about a week of elevated activity, we actually saw about a 50% increase in overall tumor burden, which, as you can imagine, in the clinical setting would make a huge difference in the context of the patients. So the next question was really to understand what mechanistically was going on here. What was causing this increase in proliferation? So to really get at that question, we used an in situ or in slice system where we genetically stimulated these slices in vitro, collected that surrounding conditioned media, and then placed them onto a number of molecularly and clinically distinct high-grade glioma cultures in vitro. And what we found here, you can see in this almost stepwise fashion, based upon the amount of elevated activity, we see this increase in proliferation in response to this active conditioned media. And this was an effect that was abrogated with the addition of tetrodotoxin, a voltage-gated sodium channel blocker that blocks the propagation of action potentials and therefore tells us that this is indeed an activity-dependent effect. So what that at least told us at this time was that there was indeed, in response to activity, a secreted factor in the microenvironment that was driving overall tumor growth, again, in a number of different distinct types of glioma. So using different types of biochemical assays, as well as 2D gel electrophoresis, we really wanted to understand what was this molecule of interest? What was the molecule that was differentially regulated in response to this elevated neuronal activity? And that gave us a short list of candidate proteins, some of which were quite obvious, which was already known to be an activity dependent secreted protein and had a role to play in glioma. But interestingly, what was actually the most differentially regulated and what came out to have the most robust effect on the tumor cells was this protein called neuroligin 3. Now, neuroligin-3 is a postsynaptic adhesion protein that sits to stabilize, the transsynaptic connections between postsynaptic and presynaptic cells. And it had never really been shown to be a mitogen in either normal or disease context. And so it was a really interesting protein for us to study. And when we alone added this protein to our tumor cells, we actually see this very clear dose dependent effect, even at low concentrations, which is really indicative of the potency of this molecule, but importantly, really tells us that neuroligin-3 alone seemed to be sufficient to elicit this, proliferative effect. Now, conversely, now that we had shown that it was sufficient to understand whether or not it was wholly necessary, we either sequestered this protein neuroligin-3 in the conditioned media by adding molecule orexin, which is its normal binding partner in the context, which sequesters the active neuroligin-3 and prevents it from acting on the tumor cells. Or we developed our conditioned media now in a genetically engineered model that had neuroligin-3 knocked out, so in the absence of the neuroligin-3 protein. And in either context, you can see that in wild type intact mice, the conditioned media continuously provided this proliferative effect. And in the absence of neuroligin-3 alone, that effect was abrogated. So this was really telling that this very important protein mechanistically involved in glioma growth. But of course, the primary question is what happens in vivo? How necessary is this protein when we look in a neuroligin-3 knockout xenograft model? And so to that end, we xenografted our patient-derived models into neuroligin-3 knockout mice. And what we found was quite unexpectedly that not only does neuroligin-3 promote glioma growth, but it seemed completely required for progression. And you can see here that in the mice that are xenografted into the neuroligin-3 knockout mice, these tumors really do not progress. And this is actually 6 months post xenograft, which is quite a while for these gliomas. So it is actually quite was an alarming finding. Now, this is in a pediatric cortical glioblastoma model. But this was actually found to be true across different brain regions, and again, across multiple different high-grade glioma types as well. So I'm showing you here diffuse intrinsic pontine glioma, as well as adult GBMs as well. So thinking about this in the context of patients, we wanted to investigate the clinical relevance of this protein. Therefore, wanted to probe its expression in a large glioma TCGA database, and found that neuroligin-3 expression is actually inversely correlated to survival in a data set of hundreds of adult GBM patients, which is really indicative of the clinical relevance of this finding, and also highlights its potential value as a therapeutic target as well as a biomarker. So to that end, we really started thinking about how can we actually clinically target this protein. And what we noticed was, as I mentioned, that this protein happened to be secreted into the conditioned media in a very activity dependent fashion. And when we looked at the peptides that we were identifying of this protein, it was clearly the peptides of the portion of this protein that sits across on the other side of the membrane. And the intracellular and transmembrane domain, as you can see here in gray, was not found at all in the conditioned media, really suggesting that this was cleaved in some type of very targeted manner. And you can see even in our Western blots, we see that we're actually detecting a cleaved version of this protein in the extracellular environment of these tumor cells. So summing up a lot of work and after trying to understand exactly what sites might be cleaved by which proteins, we narrowed this down to a small list of metalloproteases, and really found that ADAM10 was the protein that was involved with specifically cleaving this protein in an activity dependent way. So lucky for us, ADAM10 inhibitors at the time were already clinically developed. And so these inhibitors were already known to cross the blood-brain barrier to reach concentrations of a few micromolar. And so what we decided to do was xenograft, take xenografts, treat them with this inhibitor, and determine if we could really therapeutically target this ADAM10 protein in order to inhibit and limit overall tumor growth. Now, importantly, I do want to mention that in vitro, what we found was that the ADAM10 inhibition of tumor cells themselves did not seem to do much in the context of in a culture plate, which really tells us that if we did see any effect at all, it was likely due specifically to microenvironmental interactions as opposed to any type of cell intrinsic effect. And indeed, again, in multiple models of glioma that the treatment with this ADAM10 inhibitor really was found to prevent the growth of the tumor, as measured not only by overall tumor burden and IVUS imaging, but also looking histologically at the details of the tumor. So really, this really emphasizes its therapeutic relevance, and this data has largely informed clinical trials that are actively being put together. I think the pediatric glioma trial has already been launched, and the adult glioma trial is hopefully will be approved soon. And so I think this was really exciting for us because it's really the first time that this idea of cancer neuroscience in general was somehow trying to be translated to the clinic. So overall, I think these early studies really illustrate that neuroligin-3, induces this proliferation of the glioma cells by being cleaved in this activity-dependent manner by this protein ADAM10, which then acts on the surface of glioma cells. And I didn't get too much into the pathway, but we found it to recruit a number of different oncogenic signaling pathways to induce this growth. So this really puts these active neurons context of the tumor microenvironment. But interestingly, I just wanted to go back to this figure because this really informed a lot of our other work that we did. I did as a postdoc with Michelle Mange and Rob Malenka, and also that Varun and Frank have actively been working on in their lab as well. And luckily, we were thinking about this in complete parallel, which worked out quite nicely. So coming back to this model of a neuroligin-3 knockout mouse, what was still quite alarming to us was this complete stagnation in the neuroligin-3. So this result was more striking than really could be explained by the activity-regulated release of neuroligin-3 into the microenvironment, which may represent part of the mechanism by which activity may influence glioma growth, but alone really seemed insufficient to explain the magnitude of the effect that we were seeing. As most of you who work in cancers know, we oftentimes can identify very specific growth factors that might be influencing growth. And usually by taking out one growth factor alone, we rarely see such a stark effect. And so moving fully wanted to use a broader systems level kind of neuroscience technique to further understand the fundamental mechanisms here of tumor dependencies on the neural microenvironment. So digging a little deeper into this context, I mentioned that we found that neuroligin-3 does recruit a number of different oncogenic signaling pathways, the PI3 kinase pathway, the RAS pathway, but also really interestingly, when we looked at the transcriptomic data, we also found that neuroligin-3 robustly promotes the expression of a number of different synapse-related genes, including neuroligin-3, which I mentioned was in and of itself a postsynaptic adhesion protein. And so in collaboration with Mario Suva, we also went back and looked at primary biopsy samples at the single cell level of again, a number of different types of glioma patients. And what we found was that these malignant cells seem to quite highly express these same synapse-related genes, at least in a subpopulation of these cells. So these were the genes that are involved largely in glutamatergic signaling, as well as these structural synapse proteins. In this lineage hierarchy, what's again quite interesting to me, these lineage hierarchies really capture the phenotypic cellular states of these single cell tumor cells. And what we find is that the same subpopulation of malignant cells that are enriched for these synapse-like genes happen to be the cells that most closely resemble these oligodendrocyte precursor cells. Now, this was found to be true not only in Mario's biopsy models, but also in our xenograft models as well. Now, this is really intriguing because not only, in addition to the neuron synapse that we're all very familiar with, these axon glial synapses between neurons and OPCs are one of the only other synaptic interactions that have been described in the mature brain. So again, in thinking about the cellular origins of these glioma cells, we again thought to apply cues from gliogenesis and apply them to gliomagenesis. And so then we asked the question, do we see axon glioma synapses between these healthy neurons and malignant cells in the tumor microenvironment? Now, when we examined patient-derived DIPG xenografts we do see a prevalence of these synapse-like structures. And we used immunoelectron microscopy to kind of unambiguously identify these postsynaptic cell types as tumor cells themselves. And you can see this very classical synaptic cleft, these presynaptic bouton filled with vesicles, and this postsynaptic density on the glioma cells labeled here with these immunogold particles. We then use neuron glioma co-cultures to more quantitatively and precisely visualize and quantify these synapses. And we do see a number of correlations based upon presynaptic neuronal markers, as well as specific postsynaptic glioma markers. And interestingly, in the absence of neuroligin 3, such as when we co-culture with these neuroligin 3 knockout neurons, we see a marked reduction in this synaptogenesis, which tells us that, in addition to perhaps recruiting those oncogenic signaling pathways I mentioned earlier, neuroligin 3 might additionally be functioning to regulate the formation of these malignant synapses. But of course, the next question was really to understand whether or not synapses between axons in the tumor microenvironment and gliomas were electrophysiologically functional. So to test that, we used the model system where we xenographed these GFP labeled cells into the hippocampus, which was a very well-mapped circuit of the brain, and then performed whole cell patch clamping of these GFP labeled cells while simultaneously stimulating these Schaefer collateral axons that we know specifically signal input directly into that region of the graft. Now, what we find is that if we stimulate these afferent axons, these Schaefer collateral axons, while from a DIPG cell, we do indeed see that neuronal activity specifically induces these excitatory postsynaptic currents in the glioma cells that was dependent upon neuronal activity and blocked with tetrodotoxin. These EPSCs also exhibited very classical characteristics of direct synaptic neurotransmission, including this beautiful paired pulse facilitation, as well as the observance of these miniature EPSCs, which is really indicative of this single quantal vesicular release from that presynaptic bouton that can then be detected by these gliomas. And so this really, just to be 100% sure that this really was in line with true direct synaptic neurotransmission between these healthy neurons and malignant tumor cells. The synapses also exhibited a lot of electrophysiological properties that were in line with AMPA receptor dependency. And indeed, the AMPA receptor blocker NBQX, as well as the more specific AMPA receptor blocker called NASPAM, which is a calcium permeable AMPA receptor blocker, was able to block these EPSCs, suggesting that these neuron glioma synapses were indeed mediated by these calcium AMPA receptors. And I just wanted to mention that at the time we were very weary of what our data was showing, that Frank and Varun had found at the same time that this was a similar phenomenon in adult gliomas, which was really quite reassuring and tells us that these malignant cells are really hijacking this neural microenvironment in very similar ways. So interestingly, we also observed a second type of activity-dependent depolarization that had a much longer time scale. And taking a closer look at the morphology of these cells, we actually noticed that we saw these kind of spike forms that was very consistent with a response to neuronal population firing. And so further, we did find that these potentials, these depolarizations seem to scale with extracellular field potential directly, and thus was likely influenced by extracellular potassium in the microenvironment as a result of neuronal firing, very reminiscent of what we know to be true of astrocytic responses. And so this was confirmed with direct bath application of potassium alone, which induced this glioma cell depolarization, and by the addition of barium, which is an ion that can block these inward rectifying potassium channels of these glioma cells. And so overall, what we found is multiple different electrophysiological profiles. And though we haven't done, we talked about extensively earlier this week, potentially doing patch-seq to really identify which cells are associated with which electrophysiological profiles, our best guess is really coming back to this lineage hierarchy and thinking that these fast excitatory postsynaptic currents perhaps most closely correlate with cells that look like these oligodendrocyte precursor cells that in their normal context can respond respond synaptically, whereas these slower currents that look more like astrocytic currents may indeed be falling along this line of the astrocytic lineage cells. And we know that these cells are very plastic and that they can change. And so what's really important here to note is that we did see all of these different electrophysiological profiles in all the models that we tested, but we did see them at various frequencies, and the proportion of different types of electrophysiological responses was based on the model. So I think there's a lot to understand really still about tumor heterogeneity and electrophysiological responses and what function each one of these responses is, how it's getting interpreted downstream. But it's really exciting in thinking about the electrical component of these tumors. Now, to visualize this electrical activity in the glioma cells, we express this genetically encoded calcium indicator called GCAMP6 into our tumor cells. And again, these cells were then allowed to engraft in the same region of the hippocampus. And we then use 2-photon microscopy to visualize these calcium transients, specifically in the tumor cells, in response to neuronal stimulation. And indeed, what we found in response to this neuronal activity, we do see these clear evoked calcium transients that were specifically blocked by the addition of tetrodotoxin in a number of our glioma models. Of course, we also see spontaneous neuronal activity in the brain, and thus we're also able to see a large amount of spontaneous activity in our glioma cells. Now, this is clearly quite a particularly active region of the tumor, but I always like to show this video largely because it's a clear representation that all of the green fluorescence that you see here is true the current and ion flux that is happening across these tumor networks here. And so it really shows that these tissues are indeed electrically active, and moving forward, I think incorporating this novel aspect of disease pathology into therapeutic strategies is going to be really crucial for effective intervention. Now, just taken from stills from that video, you can see that the transients really seem to be propagated through these large interconnected tumor cell networks with synchronous spikes in calcium in individual cells, which you can see here plotted on the right. And so thinking about Frank's work previously describing these tumor microtubes in adult gliomas and these massive tumor cell networks, we hypothesized that these pediatric gliomas were similarly connected through gap junctions to form larger tumor networks that were being used to propagate these signals. So indeed, the addition of a gap junction blocker not only blocks the propagation of these transients, but it also greatly abrogates the amount of currents being sensed by any individual cell, which tells us that these gap junction mediated currents may in fact to amplify current across these massive tumor cell networks. So coming to the functional relevance of all of this, why might this be important in the context of gliomas? To really understand that, we thought about these normal neural precursor cells, which rely upon appropriate membrane depolarization for their own development and proliferation, and hypothesized that something similar might be going on with these gliomas. So to test this, we now again turn to optogenetics, but here we actually express these light-sensitive typically in the tumor cells themselves, as opposed to the neighboring neurons, which I had shown you previously in the talk. So these opsin expressing tumor cells were then xenografted again into the mouse brain, and we use in vivo optogenetic stimulation to determine how direct membrane depolarization affects these tumor cells. And what is like their normal neural precursor counterparts, membrane depolarization of the gliomas very robustly promotes proliferation and growth, which now tells us now that all of these different mechanisms of depolarization and electrophysiological profile indeed are important to the overall progress and communication of the tumor. Now, conversely, we're able to block this glutamatergic neurotransmission by expressing a dominant negative version of the AMPA receptor that I mentioned to you earlier, mediates these neuron glioma synapses. And when we do that, we can see that there's a stark inhibition of this pediatric glioma growth. And moving to a survival model where we again use similar constructs, we found that overexpression of the AMPA receptor actually drives tumor growth and accelerates mouse lethality, essentially, whereas expression of this dominant negative version of the AMPA receptor construct slows growth and extends mouse survival. Now, importantly, you can see that though all of these mice that did express this dominant negative version of the construct did end up being sacrificed. But you can see that at the time of lethality, almost all of the tumor cells that were left in the mass of the tumor either turned off that construct or did not express it to begin with. And so that's really interesting because it really suggests that there might be an important evolutionary advantage for the cells that can participate in this synaptic synaptic excitatory neurotransmission. So taking it a step further, we really wanted to integrate this idea of a feedback loop. We know that neuron glioma interactions are bidirectional. And until now, I have told you how neuronal activity can really influence glioma growth. But we know that reciprocally, gliomas can also reciprocally affect neurons as well. And it's been known that neurons can either secrete glutamate, they can secrete glutamate, they can secrete synaptogenic factors, and they can even reduce the number of interneurons in the microenvironment to increase excitability. Now, we found that indeed was a similar phenomenon in our preclinical models of pediatric glioma, where we see this hyperexcitable environment, in our preclinical mouse models. But really thinking about this in the context of the patient, in primary human glioblastoma, we teamed up with Sean Harvey-Jumper, who's a phenomenal neurosurgeon at UCSF, and he was able to perform electrocorticography in these adult patients just prior to resection. What we found is that in tumor-infiltrated brain, just outside of the core, we see that there's a clear increase in this high gamma band power, which is directly correlated with increased neuronal activity. And so this is really interesting because it really tells us now that gliomas are able to alter and remodel their neuronal microenvironment. And this hyperexcitability that they induce is able to potentiate all mechanisms of glioma progression that we have talked about. So both these ideas of paracrine signaling mechanisms, as well as postsynaptic currents, as well as these slower potassium-dependent currents. And so as we begin to really understand exactly what's happening here, I think it's important to really understand both of these bidirectional mechanisms in the context of a feedback loop. So moving forward, I think all of this data has really made it clear that attacking this new angle of glioma progression and interaction is really crucial to thinking about novel therapies. And I think in Heidelberg, Frank, Varun, and others are really doing a great job about thinking about this. This is a wonderful figure from one of Varun's that really illustrates that attacking these interactions from multiple ways is really what's going to be necessary. So we can think about not only these synaptic interactions between these tumor cells and these neurons, but also, tumor-tumor connections, neuronal hyperexcitability between neurons, as well as, paracrine signaling effects. And so there's lots of different angles with which, we need to think about kind of disrupting these connections and these networks that I think is going to be functionally very important moving forward. And so we've tested perampanel and meclofenamate, both clinically available kind of neuromodulatory drugs that function to slow growth in glioma models. And so, of course, further work is clearly ongoing by multiple groups now, really trying to determine the broad applicability of these drugs and these neuromodulatory agents that we already have, because that really gives us the ability to now repurpose potentially already existing drugs for the treatment of this very devastating disease. So what we now know, just to summarize all of this work in the glioma model, is really thinking what we know is neuronal activity drives tumor growth, both through paracrine mechanisms of signaling, as well as through these direct electrochemical synaptic interactions between these two cell populations. So we now know that this electrical property of cancers is quite fundamental to their progression and in facilitating disease progression. And so I think it's a really exciting time to start thinking about how we might reframe our understanding of a wide variety of malignant tissue, essentially. So of course, there are still several questions to follow up in the context of gliomas. Our lab is really interested in understanding the relevance of this phenomenon to other cancers outside of the context of gliomas. So I'll share some of the work that we have been doing now, both in the context, as I mentioned, of brain metastases, but also in the context of outside of the brain. So until this point, I have largely focused on primary brain cancers that originate from cells within the brain. What we've really wanted to know is whether non-glial malignancies from organs outside of the brain that often end up in the brain in the context of brain similarly are capable of hijacking this neuronal microenvironment and reliant upon this neuronal input. Now, this question represents a serious clinical unmet need, as brain metastases actually represent the leading cause of cancer-related morbidity, and altogether actually occur 10 times more often than all primary brain cancers combined. So this was actually quite a striking statistic that I had learned, and I think the studies in the field have been quite difficult till date, largely because of differential modeling, different organs, and so it was really an exciting part to jump into. So in thinking about the role of neurons in the context of brain metastases, of course, there has been some work that's already been done in the field. Most notably and most recently, Doug Hanahan also similarly co-published with all of us that breast cancer brain metastatic cells can actually sit perisynaptically. And though he didn't really look at direct synaptic interactions, he was able to see that they were able to uptake glutamate from the synapse and use that towards depolarization of those cells that then drove proliferation, again, in a way that's very similar to astrocytes to promote their own growth. So in thinking about other tumors that specifically may benefit from this neuronal activity, we've been studying small cell lung cancer brain metastases. So we chose this model for a number of different reasons, largely because small cell lung cancer is an extremely devastating disease. Lung cancer is currently the leading cause of cancer-related morbidity, and small cell lung cancer is a subtype of all lung cancers that has one of the worst prognoses. It only accounts percent of all lung cancers, but it specifically has an extremely high rate of metastasis, and specifically tends to go directly to the brain. So thinking again about normal development and taking cues from, its precursor cells, we know that these small cell lung cancer cells originate from pulmonary neuroendocrine cells of the lung, and thus they have an extremely neural phenotype and gene expression profile. So this idea that there may be neural gene expression programs that have some relevance to driving tumor progression is one that's been in the field for quite some time, but it's really never directly been mapped. And so to model this, we used a combination of different tools. We used genetic models, which are most commonly used in the small cell lung cancer field, which is a triple knockout genetic model that was done in close collaboration with Julian Sage. And unfortunately, though these models do spontaneously develop tumors in the lung and have a very clear course of tumor evolution and metastasis to the spleen and to the liver, they tend not to go very reliably to the brain. And so to probe these interactions in the brain, we also used xenograft intracranial injections directly into the brain of either patient-derived small cell lung cancer cell lines or these murine cells as well. So what's important to note is just as soon as we started studying these, I think it was very clear that just looking under the scope, we can see how neural these cells really look. They have a very clear neural phenotype. And you can see these long, almost look like axonal protrusions with these dendritic-like spines. And so in the literature, it's actually been shown that metastatic lesions of small cell lung cancer cells actually upregulate these neuronal gene expression programs. That happens to correlate with shorter survival and more metastatic disease. And so using immunofluorescence staining, we looked both at animal models of small cell lung cancers here on the left. These are preclinical xenograft models, as well as patient metastatic tissue from human samples. And in both cases, what we found was clear interaction between these neuronal cell bodies labeled here in white and these tumor cell processes. Again, a suggestion that perhaps in addition to structural and anatomical proximity, there may be some functional relationship here. And indeed, again, look at human patient tissue, this is really in the context of the true tumor microenvironment. And what we can see is that as opposed to this nodular growth pattern that we tend to associate with a lot of these brain metastases, we actually see quite extensive infiltration of neurons into the core of the tumor. And moreover, when we analyze the proliferation index of these malignant cells that have come in close proximity with these nerves, we actually see a significant increase in proliferation in the tumor cells that are in close proximity to these axons. And so functional relevance of these interactions, we decided to dig into this a little deeper and apply some of our past technologies to this now small cell lung cancer model. So to directly probe these interactions with neurons and small cell lung cancer cells, we turned here to in vitro co-culture systems. where we isolated our primary cortical neurons and cultured them together with our malignant small cell lung cancer cells. In this case, this is our murine cell line. And what we see is that there's a strong influence of neurons and neural activity on the proliferation of these cells, again, very specifically dependent on activity, as the effect is largely abrogated with the addition of tetrodotoxin. This was then repeated in a line derived from human small cell lung cancer samples. And what we see is that there's a very similar phenomenon in our human samples as well, suggesting that, what's quite evident is that the neurons in the tumor microenvironment can really affect the growth potential of these cells. So coming back to our in vivo models to explore this functional interactions under physiological conditions, we again turn to our same optogenetic techniques that we used earlier in our work with our interactions, where we specifically express these opsins again in the neighboring neurons, xenografted our tumor cells, and then used optogenetics to elevate neuronal activity to determine its functional relationship with these small cell lung cancer cells. What we found is that upon optogenetic stimulation and therefore elevated neuronal activity, we see that there's a clear response of these tumor cells in vivo, and there's a very strong increase in the proliferative index of these cells. And what I would just want to note is that, the baseline proliferation of these small cell lung cancer cells 40%, which is quite high in the context of tumors, which is why this is really such a devastating disease. It's known to be quite a highly proliferative cancer. But in the presence of this elevated activity, the proliferation rate goes all the way up to about 65%, which would make a huge and meaningful difference in the context of patient exponential tumor burden. So I think trying to disrupt these interactions is going to be quite functionally relevant moving into the future. But coming back to this idea of metastatic ability and invasion, what we see additionally in the tumors of these mice that had been optogenetically stimulated is that these tumors actually look far more diffuse, essentially, compared to their control counterparts, with more migration of these cells outside of the tumor core, again, in response to that elevated neuronal activity. And indeed, again, when quantified, you see there's a clear increase in this invasion capacity of the cells that are exposed to this elevated neuronal activity, suggesting that there is a significant functional impact of neuronal activity on these small cell lung cancer cells, progressive and metastatic abilities. So in thinking about, what mechanistically might be happening here, how are these neurons really affecting these small cell lung cancer cells? This is a question that we're still actively working on, but what we started is just by noticing their morphology in our co-culture models. So in the presence of neurons, what we very clearly noticed was that there seemed to be some type of morphology shift from what I showed you earlier, these long axons and almost dendritic-like spines, to these cells that had much shorter processes, seem to be much more clustered, potentially connected through these connections, and just far less branched. And indeed, when we did this quantification, we did see that in the presence of neurons, we do see a shorter distance to its nearest neighbor, shorter process length, and significantly less branching. Now, I didn't put the data here, but I'll just note that this was quite fascinating to us because this is actually an effect that was in direct opposition to what we see in our glioma models. So in the glioma models, I think Frank and Vern have seen a very similar effect, that in the presence of neuronal activity, we actually see longer processes and more branching and connectivity between these tumors. And so I think this was quite fascinating. And looking at the level of RNA-seq, we also see similarly that, when we look at astrocytic gene expression signatures, we see an upregulation of this astrocytic gene signature, suggesting that there might be some type of shift occurring here, at least in a subpopulation of cells from this very neural phenotype to this very astrocytic phenotype. And in particular, what we saw a difference in is these synapse-related astrocytic genes. So things like thrombospondin, glypican, SPARC, and SPARKLE, all of which are known astrocyte genes known to promote synaptogenesis. And so in thinking about this idea that clinically speaking, I'm sorry, my computer is frozen, there we go. Clinically speaking, a lot of these metastatic patients also present with seizures just like their glioma counterparts. We started to think about this astrocytic phenotype in the context of normal neuron and astrocytic relationships. And Ben Deneen and Sean Harvey-Jumper have all done some wonderful work really illustrating this reciprocal effect of brain cancers on neurons, inducing hyperexcitability and neuronal remodeling. And given this increase in these synaptogenic genes, what we did was we used microelectrode array systems to record from our co-culture systems and understand exactly if there was a shift in the phenotype of these neurons that we were interacting with these small cell lung cancer cells. And indeed, we do see that there are signs of elevated neuronal activity and hyperexcitability, specifically in neurons that interact with these small cell lung cancer cells. This is in vitro data that I'm showing you here, and I don't have the data today, but we just did analysis of in vivo, neurons in small cell lung cells, and we see a very similar phenomenon. So additionally, in looking at the RNA-seq data, we also saw an upregulation of a number of these NMDA channel ion genes. And so that was very highly reminiscent of what I reminded you of earlier, Doug Hanahan's work in breast cancer metastases, where these cells use perisynaptic glutamate signaling to allow for membrane depolarization and therefore growth. So again, using our in vitro co-culture models to assess whether we saw something similar happening here, we used MK801, which is a NMDA receptor, and found that this proliferative effect that we see in the neurons can be at least partially abrogated in the presence of this NMDA inhibitor, suggesting that we might be able to target these specific ion channels to block these interactions between neurons and these small cell lung cancer cells. But taking an even more broad look at some of these ion channel genes, we actually find that in addition to this NMDA signaling, there's these top rows, are the with neuron conditions. And you can see that there's a number of different ion channel class genes that are additional regulated, including AMPA channels and GABA channels and a number of different ion channels that we weren't exactly expecting. And so to more carefully understand the electrophysiological interactions with these hyperactive neurons in the small cell lung cancer brain, we used a similar paradigm that we did with the gliomas, where these cells, GFP positive SCLC cells, were xenografted in the hippocampus, and then we used electrophysiology to really understand what might be happening here. And we did this in collaboration with Bernardo Sabatini's lab and found that, again, similar to the gliomas, we were seeing multiple profiles, which is likely due to the heterogeneity of our small cell lung cancer cells. So we do see these slow evoked currents that perhaps are very reminiscent of astrocytes and are very in line with a shift to a more astrocytic phenotype. But interestingly and quite unexpectedly, we also saw these spontaneous EPSCs that are specifically in a different subpopulation of these small cell lung cancer cells, perhaps suggesting that there is still a population of cells that is highly reminiscent of neurons and is really integrating into the network, very similar to glioma. But the idea here, nonetheless, was really just this idea that these small cell lung cancer cells really were electrically active and were responding to these electrical cues in the tumor microenvironment. So to visualize this, we again turned to our GCAMP inhibitors, our GCAMP indicators, expressed those in our small cell lung cancer cells, and looked in our hippocampal slices using 2-photon imaging to see how we can visualize these calcium transients. And again, what we saw similar to our electrophysiology was that we see these spontaneous calcium transients as well as these evoked calcium transients, specifically in response activated neuronal activity, both of which that were able to be blocked with the addition of tetrodotoxin. And so what we're really gleaning from these studies of electrophysiology or calcium imaging or ion channel expression genes is really for the first time that despite not originating from cells within the brain, these small cell lung cancer cells are indeed electrically active and have the capacity to interpret input from the neuronal microenvironment. So again, to functionally understand the role of this depolarization, we similarly express our channel rhodopsin genes in the tumor cells themselves into the small cell lung cancer cells. Those opsin expressing tumor cells were then xenografted into the mouse brain. And then we used in vivo optogenetic stimulation to see what the effect on the overall tumor burden would be. And we found, again, that similar to the gliomas, membrane depolarization results in a clear increase in overall tumor growth, suggesting that this neuronal activity driven mechanism of membrane depolarization of malignant tumor cells in a non-primary brain cancer is still capable of driving growth, which is quite exciting. And so what we know now is that really there's clear neuronal infiltration into the tumor that induces progression and metastatic growth. This neuronal activity is able to evoke currents and calcium transients in these small cell lung cancer cells. And reciprocally, small cell lung cancers can remodel the tumor microenvironment to promote neuronal excitability. Now, of course, there's a lot of still outstanding questions in the context of, understanding direct synaptic interactions, paracrine signaling mechanisms, and specific ion channel may mediate this depolarization, all of which are, ongoing, in our lab. But I don't have too much time, but I just wanted to end with, another part of the story, which is that we just got very interested at the same time in really understanding, as I mentioned, how neurons can affect these tumors outside of the context of the brain. So peripheral nervous system activity has already been shown to be important in the context of development and regeneration in a number of different organs outside of the brain, both through neurotrophic factors as well as through neurotransmitting. And additionally, it's also been shown through a lot of amazing work by Tim Wang and Erica Sloan that the peripheral nervous system can play a very strong role in the malignant context as well. And various cancers, including gastric cancers, skin cancers, prostate cancers, pancreatic cancers, breast cancers, have all been shown to be largely dependent upon this innervation. And so after demonstrating that neuronal input was very influential in small cell lung cancer brain metastases, and given that neuronal activity is known to mediate the development of both neoplastic and non types outside of the central nervous system, it really led us to wonder whether innervation, specifically innervation through the peripheral nervous system, might be playing a role of influencing small cell lung cancer growth in the lung. And so to that end, if we looked at primary lung tumor samples and did find that in the human context, a number of human patient samples, that these cells do similarly express and upregulate a number of these different ion channel genes, suggestive that even in the primary context, they are capable of perhaps interpreting these neuronal cues. Now, we also looked at innervation of these primary tumors and of a variety of different neuronal subtypes, and indeed find that not only is the lung clearly innervated, but is specifically and especially in regions of the tumor. So we then asked, how might we modulate this innervation? How might lack of this innervation affect overall tumor growth? So here we were able to harness the power of these genetic models I told you about earlier. So these genetic models spontaneously develop primary tumors in the lung and eventually do a very specific time course to other organs of the body. So what we did here is just prior to tumor formation, we performed a unilateral cervical vagotomy on these mice and then used longitudinal IBIS imaging as well as histology to monitor overall tumor burden and survival in response to this lack of innervation. Now, specifically in the context of the lungs, what we discovered is that we see a very significant effect on the primary tumor burden here. And you can see here that in sham manipulated animals, almost the entirety of the lung with tumors, it's almost completely encapsulated. Whereas in the sham mice, we can barely see any tumor burden, if any. And so that was actually quite an impressive finding. And in the context of metastasis, as I mentioned, there's a very well-established course of disease progression here. With the first set of metastasis being to the liver, we actually saw absolutely no metastasis in the context of these tumors, even 6-7 months post vagotomy. And so what we know is that innervation now seems quite crucial, not to tumor initiation, but also to metastatic outgrowth. In correlation, of course, looking at overall growth and survival, we also find that this vagal innervation is critical to overall small cell lung cancer survival and progression. And this really suggests that there's real clinical opportunity here to harness the power of these neural interactions, both in the context of primary, but also in the context of secondary disease progression in small cell lung cancers. And so in the future, we really aim to further study the detailed mechanisms that are involved in metastatic progression. We're also, using a lot of tools now to really understand the role of the peripheral nervous system in a variety of cancers outside of the brain, and hope that the, inevitable outcome is really that, by studying a lot of these different interactions and pathways, we hope to uncover novel targeting strategies that are able to disrupt these very critical malignant neuron interactions that allow for disease progression across a number of different cancer models. So with that, I'll just end by just sharing that, of course, cancer neuroscience is really fascinating field, and we've really been able to develop to understand a lot about this. And I think probably maybe everybody who's participating in the seminar has ended with this slide that really encapsulates what we know about cancer neuroscience. But I think it's really nice to kind of put everything into context, is that, we know that there are these electrochemical neural cancer interactions, at least in the context of gliomas, but, do other types of cancers engage in these similar synaptic communications? Can we identify other neuronal subpopulations that might similarly be participating in these kinds of direct interactions between malignant and tumor cells? We also know that paracrine neural cancer interactions exist, but though we know that, there are still a number of questions that remain. What are the distinct mechanisms and molecules and proteins that may be at play in distinct tumor types? This might be direct effects on the cancer cells, but it could also be indirect on either stromal, vascular, or immune cells. We know that there's a clear axis of communication of neurons with other cells in the microenvironment as well. And reciprocally, how do these tumor cells affect neurons in the tumor microenvironment? We think that, we just had a meeting this week, and it was very clear that it's highly possible that different neural populations may affect tumors differentially. Some may be growth promoting, some may be growth inhibiting. And so I think teasing apart those mechanisms is going to be very important moving forward as we try to take these things into the clinic. And lastly, of course, is really understanding these bidirectional systemic interactions between the nervous system and these various malignancies, which I didn't have a chance to cover today, but I think is very interesting in thinking about, what are the other interactions that may be at play here? Are there neurotransmitters, hormones, cytokines that are at play between the nervous system and other organs of the body that can inform one another, that can coordinate in their instructions to fuel tumor growth? And so, broadly speaking, I think what's really important to know is that given the nervous system's ability to broadly regulate these tumors across numerous cancer types, which is what we know now, it's really thinking about this burgeoning field of cancer neuroscience and neuro-oncology beyond just the central nervous system and the brain, but really taking it to tumors outside of the body and really understanding the peripheral nervous system in a way that hasn't really been looked at, either developmentally or in the context of malignant disease. And so I think understanding how cancers both in and outside of the brain co-opt these mechanisms of neuroplasticity for their own growth may lead to a lot of innovative clinical opportunities and novel treatment strategies moving forward. So I think all of our collaborative research moving forward is going to be very exciting in the future. So with that, I will just end by thanking, of course, all of my lab members. Sol Savchuk drove a lot of the lung cancer work, so I really want to highlight her. Michelle Manje, of course, who continues to be a wonderful mentor and collaborator. Julian Saj, us now, Mariella Philbin's lab, the Sabatini lab, and of course, all of my funding sources. And the patients and families whose donations, without which we couldn't have done all of this research, given that we're working with primary patient samples here. And for those of you who are interested, as a small plug for my lab, please reach out if you're interested in collaborating or joining. And with that, I'm happy to take any questions. Sorry, I barely left any time for questions.
Yes, thanks a lot, Humsa. It was a great, fascinating talk. I think covered the whole range from the brain to the brain metastasis, so primary brain, secondary brain tumors to extracranial cancer neuroscience. So it was, I think it's clear that this is an emerging hallmark of cancer, and I think we this beautiful work here. And maybe I start off with for the last couple of minutes with two questions that I have regarding your brain metastatic research. So wondering about whether you think these are different populations that contribute to the neuronal hyperexcitability and the ones that again receive synaptic input, and what are your thoughts on how these two these potential populations interplay, or what are your thoughts on that?
Yeah, that's a great question. And one that I think is important, not only in the metastases, but also as we think about the glioma models. I think it's very confusing. As you know, the electrophysiological profiles of these cells, sometimes you can see them in the same cells, sometimes you see them in different cells. I think the way that we oftentimes think about it is, largely in terms of cellular state. And I think that it's very likely that dependent on the cell state, as I mentioned, like, whether or not they're more in a stem-like phase, they interact with their microenvironment and with their other tumor cells very differently than if they're in more of like a glial differentiated astrocytic state, for instance. That doesn't necessarily map to small cell lung cancers, but I think in terms of what I'm trying to say is really just, I think that they are separate phenomenons. That being said, because these cells are so plastic, and Mario Suva's work and a lot of other people have shown that these cells, plasticity is a very important part of a lot of these malignancies, and cells are oftentimes easily moving from cell type to cell type. I think it's important to consider that, there might be an intermediate phase where they're participating in both. And so the way that I like to think about it, and perhaps it's a little too reductive, is really this idea that, the astrocytic kind of arm are really the ones that are really, playing into this hyperexcitability, synaptogenesis, excess release of glutamate and things like that. Whereas these more stem cell populations are maybe more integrated into the neurons and just like responsible for receiving more input and spreading it to their neighbors. I think more work to be done here. I think there's a lot of, sorry, I can go on.
Yeah, I think that sounds very interesting. I think it sounds like teamwork between different cell states that really drive progression together with neurons. I think that's a fascinating concept. I think that one that we should definitely, or that the field should for sure explore. I think that makes a lot of sense, also bringing these fields together. And another very short question, knowing from your work also that you looked at breast cancer brain metastases where neuroligin 3 did not play a role. I'm wondering again about the small cell lung cancer. We have this more neuronal phenotypes, and wondering, do you believe that this could potentially play or could be distinct between different brain metastases entities, or do you think it is simply completely different between primary brain tumors and brain metastases?
That's a great question. I had the same thought, because the breast cancer cells, at least specifically in response to neuroligin, didn't seem to have a very, very clear response. I do think that it's very subtype specific. And I know, and it's very clear now that small cell lung cancers, as you mentioned, are very neural in their phenotype, since they do express the neuroligins at quite a high level. So I wouldn't be surprised if neuroligin might be playing a role in the small cell lung cancers. I don't know as much about the breast cancer realm and what their baseline levels of activity and expression are of a number of these synaptic proteins and ion channel genes and things like that. But I also think that it's a high possibility that they are subtype specific. And so, we tested one model of one type of breast cancer brain metastases, but, dependent upon, the subtype of that type of breast neuronal, perhaps there are ones that have a higher propensity to get to the brain. And so I think, really teasing that apart is going to take a little bit of work, but I do think that there's probably not one conserved mechanism across all of these cancers in the brain. And then we're going to have to tease out something a little bit more difficult in terms of the detailed mechanism across each.
Okay, thanks. So Jeremy?
Oh yeah, thanks. It's a really great talk.
Yeah.
I had just a general question about the role of synaptic input or electrical activity on glioma or small cell lung cancer growth in the brain. As you demonstrated in glioma, the electrical activity is essential for the growth of the glioma tumors. I'm wondering if this growth is dependent on the electrical activity, or is there, in the normal brain, if there isn't synaptic activity in a circuit, a lot of those redundant circuits are eliminated. And I'm wondering if there's some active sort of destruction of inactive tumor cells, like hey, this neuron's firing a lot, we need to support this metabolically. And similarly in glioma, is this glioma cell showing a lot of activity? We as other members of the tumor microenvironment need to support these cells metabolically. And is the reason that they're not progressing when you eliminate the electrical activity due to active elimination of the tumor cells, or simply lack of a growth signal, if that makes sense?
I'm not sure that I wholly understand the question. I'm not gonna lie.
Yeah.
But I will say that I think perhaps some of the work that maybe Frank and Vern have done, where they can specifically like monitor one cell over time and even ablate different cells around it, might be a way to test that hypothesis, to really understand exactly which aspect of this is going to be more important. I think there's still a lot to understand in terms of, if I understand you correctly, whether or not it's the neurons that are supporting a very active cell, I guess, or whether or not this cell is like actually receiving. I think the two go hand in hand is kind of, but I think I perhaps didn't understand your full question.
Yeah, I was sort of figuring out the best way to convey it while I was talking. So, yeah, it was basically, you know, is the lack of growth a result of just there's no more input to the glioma cells because you have this dominant negative, or you have this gap junction inhibitor or something, or is there active removal of the glioma cells by the immune system? You know, like are they being killed, or is it simply lack of a growth signal?
Yeah, that's a great question. So I haven't looked into this in great detail. My feeling is that it's lack of a growth signal. It's really just a cue that they're receiving from these neurons that then, you know, downstream, we know, you know, that elicits a lot of changes downstream in signaling pathways like kinase pathway, for instance, and like that drives growth. And so the blockade of that signaling pathway, therefore, essentially stagnates growth. If it was elimination, that would be even better, to be honest. But I think we're still working on that component. And I think, of course, as you mentioned, there is, you know, I wish I could have gotten into like the big neural immune component that is definitely at play here and is clearly an elephant in the room that we have to address simultaneously as we interact all of these systemic interactions. So I'm sure there is something at play there as well. And if we block neuronal activity, perhaps part of it is that we're able to then activate a whole different of immune cells as well. But I think there's a lot of different people looking into that right now. And so I think really understanding the interplay between all of these will be important. But specifically in thinking about the neuron tumor context, I think it's a growth cue.
I see. Cool. Thank you.
Mm-hmm.
All right. Frank?
Yes, thanks a lot. Humsa, for this fantastic talk, really spanning almost the entire large parts of the arena of cancer neuroscience. And I think it makes a lot of sense, of course, to focus on small cell lung cancer. I mean, as you said, I mean, this is a cancer that is very neuro-like, right? It's the most frequent neuroendocrine cancer of the lung. And also in cancer entity, that now that we leave again this habit of preventive prophylactic whole brain radiation, we see more and more now again in the clinic of these of metastases. So it's kind of, this is probably also so now we need to know better. Again, better know that we leave this harsh neurotoxic measure. We need to know better how to better treat them. So my question for you is I mean, that you start, of course, with a very neuronal-like cell type that is then transforming interestingly into this astrocyte-like type. That's quite interesting. So what would be your kind of assumption or hypothesis, or maybe you even have data on that? So what happens when you start with a very non-neuro-like cell type, like a carcinoma, right? That is not, has not these neuronal features to start with. So what would happen then? Would you
what happened?
Different changes, or what are your ideas on that?
I mean, that's a great question. I think that it's very, very tissue dependent, and that these aren't going to be vast things. And we know that largely because even just in the gliomas, we know the gliomas do not necessarily behave in the same way. I think specifically when the small cell lung cancers, in thinking about the metastases to the brain, I think it'll be interesting to look at there might be some specific conserved mechanisms, because, it is also possibly enter the brain. The neurons are already, they just need to find a way to harness the power of the neurons, and therefore, a subset of them turning more astrocytic might be more helpful. And I think perhaps in terms of, taking on essentially taking on the phenotype of the host organ that they have now colonized is very important. And so I'm not necessarily surprised that, there is maybe a subpopulation that looks very neuronal. There's a subpopulation that looks very astrocytic, and they're almost kind of mimicking the normal microenvironment of the brain in their own community, essentially. I haven't looked in detail as to, I think what, to start with, I think what's going to be really important is really thinking about just the differences even in the primary versus metastatic context. And so doing some what the sequencing that we have done is from the metastases in the brain, but I think even starting from the primary sample paired with the metastatic sample is going to be really important to think about how those phenotypes similarly changed in both contexts, because I think my gut is the organ of colonization is really important, because I think, as we all know, of course, what's driving a lot of these, cancer cues and really modulating and remodeling them to shape however they need to survive and thrive in specific environments. And so I think we don't yet have great data on that. We are we're currently trying to do the primary sequencing right now. So I'll keep you posted on that, but it's a great idea to start looking at, carcinomas and things like that, just to see what might happen. But my feeling is that it will be different dependent on the tissue of origin to begin with and the tissue in which it ends up in.
Great. So we have a question from the chat from Martin Schmelz, who asked, great talk. Could you comment on your ideas whether this is just depolarization of the cancer cells to promote growth, or whether a certain on-off pattern or even synchronicity as delivered by the neurons is required?
Yeah, that's a great question, and one that I'm really interested in. We're actively trying to study that by using optogenetics to essentially kind of play in different patterns of activation and hyperactivity essentially in these tumors to see what might be going. What's interesting is that, to be honest, like everything that we've tested so far is actually at physiological frequencies. And so we test specific neuronal populations at the frequency at which they fire in a normal context, in their healthy context. But a lot of these metastatic patients, of course, they present with they present with seizures. And so what we are testing now is really understanding whether this pathophysiological seizure frequencies of neuronal hyperactivation similarly, or perhaps even more profoundly, affect tumor growth, because there must be a reason that's for the tumor in which they're creating this hyperexcitable environment, right? And so I think it's going to be really interesting to kind of tease that out. And then if we can figure that out, I think that opens up a lot of different possibilities of DBS and things like that, where, that's currently used in epilepsy that we can now then apply to some of these tumors. And so I think it'll be really exciting. And I think that's something that we're actively very interested in working on. So I hope to better know the answer to that question soon.
Right. I was just, and the idea of synchronicity, of course, is also methodologically kind of linked, right? As you induce it, an extreme synchronicity with your light pulses. So that might be a quite interesting signal for the cells as you force them to fire together, right?
Right, exactly. And you're right about that. Yeah. So we have to maybe think about how to play in and play off all of these different ideas. I think that is, also somewhat difficult to mimic, but again, showing, both Frank's work and our work we've also looked at these blockers that can play with these patterns of synchronicity, and clearly, obviously turn off synchronicity by blocking that. And that seems to have a severe impact on overall tumor growth. So I think that's definitely playing a role.
Great. Then let's end the seminar with the last question from Thomas, and then I think we are already a bit over time. So let's.
Okay, yeah, so that was a real tour de force, really fantastic data. So you also found fast EPSCs in your SCLC recordings that very much look like AMPA-ergic minis. So have you tried also AMPA receptor blocking drugs in the small lung cancer context, whether that inhibits proliferation?
A great question. We are actively working on it. I'll put it that way. I know there's another group in Germany that I think is also particularly interested in that question and definitely has tried that and seen that AMPA receptor inhibitors seem to have a very strong effect on these small cell lung cancer cells. And so that was something, the EPSCs were something that I wasn't, to be quite honest, I wasn't necessarily expecting. I was expecting some of the slower evoked currents, but then they just, these EPSCs just kept popping up in our electrophysiology recordings, so you can't really ignore them, and so they're quite prominent. But we have yet to do. We're currently working both on the electrophysiology, like using the inhibitors in the electrophysiological context and blocking those EPSCs, but also functionally in our co-culture models to see whether AMPA inhibition can slow growth the same way the NMDA receptor inhibitors do.
Yeah, and I think that will be very interesting to see whether there are interactions between AMPA and NMDA. I mean, like in the typical synapse where you have to have a depolarization action first and then unblock the magnesium block of the NMDAs and then, really suck the calcium into the cell.
Right,
I mean, that
exactly.
will be very interesting to see whether that kind of cooperativity is there as well. Right, and then also you basically stress a lot the role of depolarization, membrane depolarization. But I mean, what's in the end a transductor? I mean, what transduces membrane depolarization in glioma cells or small lung cancer, whatever is tumor cells, into the signal that is actually biologically relevant for tumor biology?
Yeah, I think that is the question, shall I say. I think we are still trying to figure that out. And I think, Frank's also doing great work on really trying to kind of decode these signals and really understand exactly what the correct messengers might be and exactly what that means downstream. And I think answering that will really help us obviously therapeutically speaking, but I don't have the answer just yet, because it seems like a number of different ion channels, a number of different ions itself can seem to elicit this effect. That's why I very broadly call it depolarization. We have looked at some, basic, as I mentioned, kind of MAP kinase that we do see very specific, almost expected pathways that are, getting phosphorylated, getting upregulated, that they're known cancer drivers. But what might be that if there's a specific and, specific interpretations in different glioma cells based upon different cues or different mechanisms of depolarization, different ion channels, different patterns of activity, all of those things we don't necessarily know. But I think it'll be really interesting to think about.
Thanks.
So thanks again, Humsa. Thanks to all participants for the lively discussion. And I think then we're off to next week's talk, where we also will again announce via Twitter. So thanks a lot to everyone and have a good evening.
Great. Thanks, everyone.
Thank you.
Bye.
Bye.